In this video, we demonstrate OoDAnalyzer. In the example, the training set is biased in color by only having white cats and black dogs. The classifier predicts the black cat to be a dog and the white dog to be a cat. These samples that are not well covered by the training set are called out-of-distribution samples. To tackle this problem, we develop OoDAnalyzer for interactive analysis of out-of-distribution samples. Our system consists of 3 views: a control panel that helps explorations, a grid-based visualization that illustrates OOD samples in context, an information panel that shows image and saliency map representations. Case 1. OOD analysis of the animal dataset. Part 1. OOD analysis in initial data. The expert first increases the cutoffs of the OOD score slider. Several areas of OOD candidates arouse the interest of the expert. The expert decides to check these regions one by one as they all contain OOD candidates. Region A mixes dogs and cats together. Zooming in, he finds many misclassified dark-colored cats and light-colored dogs. Then he selects these 2 categories for further exploration. Their saliency maps highlight the whole body. He uses juxtaposition mode to compare the distribution between training and test data. In training data, dogs are mostly in dark color and cats are mostly in light color. In test data, however, dogs and cats are in different colors. Thus, the expert confirms that lacking diversity in color between cats and dogs causes these OOD samples. Back to the overview, the expert continues with other regions. In region B, some cartoon images are misclassified as rabbits. He decides to use superposition to figure out the reason for this error. There are no similar cartoon images in training data around them, but many rabbits with prominent ears. The saliency maps also stress the rabbit-like exaggerated ears. Missing cartoon images accounts for such OOD samples. Next, the expert moves to region C. Dogs are completely confused with wolves here. He selects some images and examines their neighbors and finds some huskies. Then he turns to the training data, but can hardly see huskies. So he considers lacking huskies is the major reason for the appearance of these OOD samples. Similarly, in region D, the expert finds some tabby cats misclassified as tigers because of the black straps on their body. Few cats in training data have such patterns. This is identified as an underlying cause to have such OOD samples. To mitigate the problems of OOD samples, we successively add the corresponding missing images to training data and get accuracy increases. Moreover, after retraining the model, there should less OOD candidates in the updated layout. Part 2. OOD analysis in incoming data. New images come into test data and cause accuracy degradation. Another expert wants to make a diagnosis and reloads data into our system. He quickly recognizes 2 regions of high OOD scores. Zooming into these regions, he discovers leopards hidden among tigers. As knowing that training data includes no leopards, he identifies leopard as a new category. 400 leopards are added into the training dataset and the accuracy of the model goes up again. Case 2. Improve the classification of renal edema. Part 1. Overview. The doctor compares the data distribution between the training and test datasets. The test set has many OOD candidates. The REA and REA-free images are better separated in the training set. He decides to use the superposition mode to examine those OOD candidates in context. Part 2. OOD candidates with no obvious renal thickening. In the superposition mode, OOD candidates gather into smaller groups. The doctor starts from the boundary. Those images look like REA-free. He zooms in and finds some images with intrarenal cystoid cavities, but no obvious renal thickening. They are misclassified as REA-free. In the neighboring, the training set has no such images. Look at the saliency maps. The intrarenal cystoid cavities are not learned. He switches back to the overview and finds a similar image in the bottom left region. He zooms in and finds more such images in the test set. He confirms that liking ADMA images with intraretinal cystoid cavities, yet in the absence of retinal thickening, is a major cause to have such OOD samples. Adding samples for compensation, the accuracy is improved from 90.47% to 93.02%. Part 3. OOD candidates with hyperreflective falsi. Zoom in the bottom right region. Those OOD candidates all have HRF. Some are predicted correctly as REA, while others are not. Look at their saliency maps. The model learns the HRF and makes correct predictions only when the HRF is obvious. The doctor finds that HRFs in the training set are all obvious. So missing REA samples with less obvious HRFs leads to these OOD samples. By adding such missing samples, the accuracy is improved from 93.02% to 94.53%.